Genetic code and its properties. Genetic code. Let us know about genetic code clearly. DNA, the deoxyribonucleic acid, carries genetic information from cell to cell and from generation to generation. DNA is a double-stranded helical structure made up of phosphoric acid, deoxyribose sugar, and nitrogen bases. The nitrogen bases present are adenine, thymine, guanine, and cytosine. The genetic information can be written by different combinations of these bases. The special coded information is called as genetic code, and the coded message is called as cryptogram. The 20 amino acids are alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. A triplet code made up of 3 letters specifies 4 into 4 into 4 is equal to 64 amino acids. Therefore, there are 64 triplet codes for 20 amino acids. A group of 3 nucleotides specifying 1 amino acid is called as codon. The in vitro studies by Marshall-Nirenberg in 1961 and in vivo studies by Janowski in E. coli and man led to the formulation of the code in the form of a table for 20 amino acids. Salient features of genetic code. Triplet code. Genetic code is a triplet code, a combination of 3 nitrogen bases specifying 1 amino acid. Example: UUU accounts for phenylalanine. Degenerate code. As 64 codons are possible in a triplet code, 1 amino acid can be coded by more than 1 codon. Example: leucine can be coded by CUU, CUC, CUA, CUG, UUA, and UUG. Non-overlapping code. Code should be always read in groups of 3 nucleotides from a starting point when exposed through a reading frame. Commaless code. Punctuations are not specified between any 2 codons. Unambiguous code. One particular codon will code only for one amino acid. Universal code. Genetic code is universal, that is, a codon codes for the same amino acid in different living organisms. Starting codon. AUG acts as the initiator codon, and it also codes for methionine, so it has dual functions. Nonsense codons. These are termination codons which end the process of synthesis of polypeptide chain. Example: UAA, UAG, and UGA are termination codons called as ochre, amber, and opal respectively. Mutations and genetic code. Mutations are the sudden changes occurring in the DNA to bring about structural changes in the organism. Mutations can be point mutations, frame shift mutations, insertions, deletions, etc. Point mutation. Take an example having words with 3 letters each. Ram has red cap. Insert B before R, then there is a shift in letters as shown below. Insert I after B, the rearrangement as shown below. Insert G after I, that is, the total word big is inserted. So the sentence becomes, Ram has big red cap. Same process can be repeated by deleting the letters. R, E, D, one by one. The new sentence format can be seen. Ram has red cap. Can be written as below. Insertion or deletion of the bases in the reading frame re-alters the frame, and such mutation is called as frame shift mutation.